Is Robert Mullen. I'm the Director of Agronomy for Nutrien. So my primary responsibility is on the wholesale side of our business. And just as background, I call myself a recovering academic. I spent 7 years at Ohio State University,
I see.
soil fertility nutrient management specialist. So I'm statistician. Yeah, I love statistics. Anything that we can do to make things more complicated, I'm all for.
My taste.
And then, Kaiyu, I think you also know Austin, but I asked Austin from Illinois Farm Bureau if he would sit in today as well. So.
Morning, Kaiyu.
Hey, Austin. How are you?
Good. Are you making it through? Do you teach any courses at University of Illinois right now?
Not this semester. Not this semester. I had a teaching release arranged a long time, half a year ago. So I'm not teaching this semester, but I still involved. All the research stuff is continuing. The field work is very challenging this year, but we managed to do almost all of them. And we just cut our 2 sites in Nebraska. We just entirely focus on our U of I sites. And then so, things are mostly normal, but we are kind of battling with all these COVID in the campus situation. Yeah, you guys heard about that. Yeah.
Yeah, I think Illinois State's setting records for the number of cases every day.
I know.
Not a good thing, but anyway.
Yeah, kids, young kids like parties.
Yeah. Who thought,
Anyway.
I know, as an Illinois State alum, I'm definitely, this is not one of the times that I'm happy to see our university in the news. So
I know.
I think Iowa State and Iowa are still beating us though. So
I think it's the nature of young students, young undergrads. You couldn't really blame them, but we just hope that we can better, we have better procedures, we have better protections for everybody.
Are they using the tracking mechanism with the saliva test and that down? I heard that they're using that down to U of I. Do you think that's making a difference on your keeping things under, more controllable?
That's definitely helping. I'm seriously thinking about that, because at least our campus has a very concrete number about who, the population that tested positive. Like we really know this number. And then because of knowing this number really help protection, because now based on my understanding, all the students tested positive, they actually has been actively monitored. If they are quarantined, and then so I think university really pay very careful attention, and then also that provide a mechanism that we can track about all the students that has been interacted with these positive students, and then so I think that's definitely help. And the saliva test is actually probably the cheapest approach at this moment, and also highly robust and reliable. So I think this is an invention coming from U of I, so very proud about that. But definitely we can do more about how to make sure the campus or the town is under control for the COVID situation. Yeah. But I'm very hopeful. And then and also I think the campus leadership as well as the whole population of the campus, they showed the determination and the resilience. So campus leader can easily say that, hey, go back home.
Yeah.
That's a very easy decision to make.
Yeah, it is.
And then so, but I think they are hanging there. They are trying to make positive progress. We are, all the faculty members and students are part of that, and I think it's making very good progress. I have to say this. If the government decides that the students needs to be, go back to school, then we just do the best we can.
Yep, it might be a good model for the future if we're going to have to continue to deal with it. Looks like.
Totally. We hope we won't have a COVID situation for a long time if we get the vaccine, if we get passed by.
I'll just go ahead and Kaiyu, I'll just hand it over to you.
Sure.
You can share your screen if you need to. I think you should be able to. Yep.
Yeah. Okay. Well, really appreciate Julie, appreciate Robert, Jeff, and Austin for your time. come to spend time with me. And then so I think I'm gonna use this time to really provide an update of the project that I have with NREC, and also gaining feedbacks from our NREC stakeholders and not only committees, but the committee representing the broader stakeholder interests. So I really want to collect your feedback and the guidance to enable better research ongoing. And then so to start with, the project that I got funded from NREC dates back to 3 years ago. The project that I have is about cover crop. And then so the project that I have is about cover crop. Basically, essentially, we propose to collect the different data sets, both from the trials that NREC has been sponsoring doing, and then some of the models, and then possibly leveraging satellite data, and put them all together to understand better about cover crop. And then with the key question about how Illinois farmers or Midwest farmers in general, how we can grow cover crop to best benefit farmers' economics, so we want to make sure that cover crop, what cover crop is suitable, how we grow that, and what's the benefit of that. And then so this is the key question that we have. The approach that we are doing is not the classic trial approach. The trial approach is very, very important. And in fact, we work with Villamil, Dr. Villamil, and we also worked with the Purdue professor, Professor Armstrong, in his trials. So we try to work with the professors, scientists who already collected cover crop on the trial setting, primarily funded from NREC. But we want to make a further step by extrapolating the current results to multiple yields, to longer impact in the model framework. And I understand that part, I can spend more time to talk about that because, using this approach is pretty new. And I think communication is very important. Demonstrating it's working also very important. So I will say that a lot of the conversation that I hope to collect wisdom and guidance from our stakeholders here is how to best communicate the results to our stakeholders and how to make these NREC research investment really pay off. And then so I gotta go into details, but I will first provide an update, what we have achieved in our NREC project. And then second, I want to have time to really hear our stakeholder committee member here to give me guidance about, what do you envision the questions about your specific need about the cover crop and the nitrogen fertilizer use, because these two are the topics that my research program is really focusing on. We try to understand cover crop, try to see how we better use cover crop, as well as how to better use nitrogen fertilizer for farmer in the kind of the commercial farming settings. And then last but not least, I think communication is very important. Being a founded researcher of the NREC program, the previous few years, most of our research are primarily focusing on attending the annual event, and then we have one investment insights material produced. But beyond that, a lot of the work that we have been focusing on is to make sure we get the research done. Now research reached to really the later stage. We finished a lot of the work. I think it's really the time that we step up and start to communicate our research, and I think that's very, very important. I feel that this is the time, and we spend a lot of time to make research work, but now it's the time to communicate. And then how to effectively communicate, I really would like to select the feedbacks from you, and then to help me better communicate with our stakeholder. And then so this is a list about what we have achieved, and so I would lump them up into these four major points. The first two are the work that has been published. The third and fourth are the ongoing work that we already finished. The paper, it's under review. And so in other words, that in a nutshell, we have finished all the promised work, and I just gonna quickly go over that. So the first work that we did is we did a pretty comprehensive metadata analysis with Maria and Villamel's group together to understand cover crop benefit to soil microbiome. And so I'm gonna go into a little bit details of that. The second aspect is the research that we actually end up having investment insights produced by Mary of NREC and share with the stakeholders. So in that particular case, we work with Emerson Narzinger, the professor in U of I crop science, looking at his trials and understand, can we come up with, can we sense that crop stress or nitrogen stress in their trials using some of the satellite technology. And so that's a study already published. And all these studies are very recent studies. So you will see there, this paper published just this year, this paper published last year. And then the third and fourth are really focusing on the core question of cover crop suitability in our landscape. And then in particular, we did a whole, 3 I states, Illinois, Iowa, Indiana, 3 I states study, understand whether our crop model really work. And we will go into details because I think one of the biggest questions from our stakeholders, especially when you think about model, they will ask, does it work? Has that benchmarked? Does that reproduce reality? And then we spend a lot of time to conduct probably the most rigorous study to really demonstrating that the model actually can reproduce the impact, the patterns of crop yield as well as the carbon cycles, water cycles. Last but not least, this is a study that we focus on understand the cover crop growth condition. We try to simulate cover crop growth as well as its impact for soil organic carbon. And then we did it use at the 7 sites that Professor Maria and Villamil previously had NREC cover crop trials. And so we fully leverage these historical data that has been collected through the NREC's previous investments. And then we try to do the analysis to see whether our calibrated model can reproduce the cover crop growth, the cover crop biomass, the cash crop yield, and these aspects. And then if we can demonstrate that the model works, then we can start to ask some questions about what if we start to really simulate, grow cover crop in this landscape, in this specific site for 10 years, 20 years, what will be the implication for crop yield, for soil health, for soil organic carbon? And then this is the question that we are addressing. Both 3 and 4 are finished. The papers are submitted in the leading journal of our domain. And then so it's mostly on the kind of revision stage. So we are waiting for a reviewer to give us the reviews back, and then we address them and get a paper published. I envision both papers, I can work potentially with Mary to generate some investment insights, especially the 4th one. Because in the 4th one, we discuss about, a few details about cover crop practice, like when you put the cover crop seed on the ground, what's the implication of that? Do you think you need to put some fertilizer even in the cover crop? Usually most people don't do that. But then if you wanna try to avoid any coming year cash crop penalty with cover crop, sometimes you have to do a little bit of fertilizer. And then so we really look into kind of these details, and then I really hope to collect feedbacks from our committee. So I'm gonna go into a little bit details. So, but they all relatively stay at the high level. So this is the study that we did, the metadata analysis, really looking at the soil microbiome impact on the cover crop. And so you see the title. And then we actually reviewed about 300 papers, published or before we write the paper, before 2019. And so that's a huge effort. The student really put a lot of efforts. The short conclusion that we gain is a cover crop really help improve the soil microbiome abundance, activity, and diversity. So in other words, that cover crop helps, especially from the soil microbiome activity. It has been really demonstrated from these past 300 plus literatures and studies to demonstrating this is the case. There are some nuances that also affect the magnitude of that benefit, depends on the climate, termination methods as well as tillage. And then so for the sake of time, I will not go into the details, but this is a very nice study to support the benefit of cover crop regarding the soil microbiome activity. The next study is a study that, as I mentioned, we work with Emerson Nafzinger in their trial, and then a student of mine, who actually flying, drones, and also look at some satellite signals to see whether we capture the in-season crop nitrogen stress. And then the reason that we really care about in-season is if we can really detect early stage nitrogen stress, then that basically provide a possibility that we can side dressing the crops and to ensure that there's no nitrogen stress afterwards. So this is a study that we did, and they even have some media coverage of that. And then so going into a little bit detail, we are looking at particular sites in the South Farm of the university, and they're looking at the trial plots. These trial plots has nitrogen fertilizer applications from 0 to up to 250 pounds per acre. And then we also have different ways of applying nitrogen. So for example, there's trials that just have the standard fall spring application. Some trials have the split nitrogen application. So there's a variety of application with replicates. And then in that study site, we actually did a whole growing season data collection. So these are different dates of the growing season. The field work is done in 2017. And then at different dates, we collect drone image and the satellite image. And then we also conduct the leaf sample analysis, measure LAI, leaf area index, which is the biomass. We measure the canopy nitrogen content, as well as, counting the phenology stage. So then what we look at is showing this graph. It's a little bit complicated graph, but bear with me. It should be straightforward after I explain. So if you only focus on the left side, which is the A panel, what you are seeing here is the X-axis is the day of the year. So, day of the year after planting. So 20 means 20 days after you plant the crop. And then the Y-axis refers to the whole crop chlorophyll content, which is an indicator of both biomass as well as ability to do photosynthesis. So you will always see that as crop go up, increase their biomass, you expect to have more chlorophyll content, more photosynthesis, and actually you are seeing this. So all these trials shows goes up and down pattern. Go up means it's growing up, peak time, and then they start to senescence. Now, different colors is very interesting because different color refers to different nitrogen application. So what you are seeing here is the different nitrogen application. Red dot means there's no nitrogen application, 0 and 0. So there's two numbers. The first number is you apply the fertilizer in spring before you plant. The second number refers to whether you did the side dressing or not. In this example, we only look at the one-time application before the spring, before the planting. So you only see these numbers on the first part. So the red means it's no fertilizer. Green means 50 pounds per acre fertilizer, and the 100 in blue, 150 in yellow, 200 in black, and then 250 in purple. And then so you will see that these different colors indicating different nitrogen trials. In general, as you put more fertilizer into the ground, your yield, your chlorophyll index increase, because that's a strong indicator of biomass as well as the canopy nitrogen. So increase. And then on the very right side here, you will see that different trials actually shows different yield number. So if you don't put any nitrogen in the landscape, you still get about 150 yield bushels for the corn. And then if you do 50, it's about 180. If you do, let's say, 200 bushels per acre, 200 pounds per acre, then it's the black here. And then the purple shows the highest yield. So in other words, that if you see the satellite signals, it's pretty much can capture the canopy chlorophyll content, which is related to the crop stress and nitrogen content. And then so you clearly see that signals has been picked up by satellite as you increase the nitrogen content, you increase the chlorophyll content, and then it's corresponding to the yield. Now, what's most interesting here is if you want to know the yield impact, usually you need to wait until you harvest. So if you know these numbers, you can only get to know this number when you wait till the growing season ends, harvested. But these are all in season. So in other words, in the middle of the season, you actually start to see that, hey, there's some stress that show up in these low nitrogen trials, and it's pretty clear and statistically robust. And then if we look at the right side is only looking at the difference between different trials, chlorophyll content, minus the lowest trial, lowest nitrogen trial. So basically, what we are showing on the right side is we look at any curve difference between the curve and the red one, because red one has been chosen as the benchmark. Because we put the lowest nitrogen there. So if we look at in this way, we will clearly see that this is the difference. You start to see the difference really after 50 days of your planting. So after your 15 days of planting, if you have a nitrogen stress, use some of the technology that we proposed from Jones or from the satellite, we actually start to detect that there's a nitrogen stress that emerge. And then these nitrogen stress become more and more obvious in the later stage, which provide an indication that if you want to use satellite to detect when to put actual fertilizer on the ground, you actually possibly can do that. And then we also look at the studies that because there's trials that also put fertilizer around the different, primarily the V7 and the V9 stage. And we actually see that if you put fertilizer during that period of time, there's a small mitigation of the nitrogen stress that you can see. And then so this type of the study really demonstrate that, first of all, using satellite data and some sensing technology, you can help you to detect early nitrogen stress in season, and also that potentially provide a remedy for if you can apply the side dressing to avoid the end of season yield loss. So that's this study that we did partially funded by NREC. And then the third study, I will go into the third and fourth study. Start with the third one. The third one is really to provide a very comprehensive understanding and the validation about whether our model that we are using works. And I know it's a big question that a lot of our stakeholders actually have concerns. So we spend a lot of time to understand, can we really demonstrate that? To do that, on the left side, we show this is our model structure. And then so depends on whether you're interested in going into details, but this model actually does have a pretty complete ability to simulate crop growth, the water cycles, the nitrogen application, as well as including the microbial growth and activities. And then so it's a comprehensive model. But then as you know that all the model may have uncertainties, especially how you make sure the parameters work, how you really validate your model, to show that the model actually has ability to transfer from one location to the other. And then to do that, we look at these 7 sites that we have a huge amount of rich data set that collected. So these sites collected, I would call them the golden standard sites, mostly because these sites, they not only collect the management practice, the soil samples, the crop yield, but they also measure the carbon cycle, the carbon photosynthesis, respiration, and soil moisture over the whole growing season at a very high frequency. For example, the carbon, the photosynthesis measure, they use so-called eddy covariance measurement. You can actually get photosynthesis every half an hour. And then, so these are the sites that, in the US, they've been widely regarded as the most, gold standard data set to validate your model. So we take these sites, they are in the agriculture sites, corn soybean rotation sites. So we take these sites and then we look at, whether we can reproduce the pattern. So on this slide, on the left side is a typical site that basically we have, these sites has these so-called eddy coherence station. They not only measure accurately about meteorological conditions like weather condition, but they also measure the water flux and carbon flux at any 30-minute frequency over the whole landscape. And this landscape is the whole field landscape, usually 300 meters by 300 meters. So it's like including the whole field, variability. And then they take measures continuously over the whole growing season. And we are looking at all the 7 sites distributed in the broader corn belt, and then they all corn-soybean rotation sites. So it's all agriculture sites. And then so what we are doing is we try to look at these sites and see whether our model, after the limited calibration, can we reproduce the patterns that we care about? Can we reproduce the variables that we care about at these sites? And then so the results on the right side shows that there's a few variables that we really care about. And then so these are scatter plot, starting from the first one. This is the scatter plot. X-axis is the observation. The y-axis is the simulation. What we are showing you is the photosynthesis rate at every site. And you will see that these are the different sites name, different color refers to different sites name. And then so we simulate the model and we calibrate the model and we simulate the model. And then we look at the patterns when we don't, we basically separate the data set from calibration, from testing. We calibrate the model using one part of the data, and then we test entirely using auto sample data. And then so these are showing the test results. The GPP refers to the photosynthesis rate. The R equal refers to the whole system respiration, how much carbon has been respired. The NEE refers to the net exchange of the carbon. So essentially how much carbon has been taken by the plant, how much carbon has been losing from the respiration from both soil and the plant. And then the last is LAR, which refers to leaf area index, which you can basically envision as the biomass, background biomass. So if you look at these results, it's actually really give you the high confidence that the model it can actually pretty rigorously capture how much photosynthesis they capture at the site level, and then how much respiration you are doing, as well as the biomass information. So this gives a lot of very big confidence that we can actually make model work. And we are looking at all the 7 sites that spread out of the corn belt. And then so next, what we did is we then say that, hey, if we already demonstrate it works at the site level, can we demonstrate that it works at the broader scale? So what we did is we did this regional simulation that we look at every county in the corn belt, in the 3 I states. So Illinois, Iowa, Indiana, look at all the counties. And then we compare, we use our simulation and we compare our simulation with the USDA NASS data. And then NASS is the public county level data that collected by the USDA from the survey. And again, when we do this validation, we separate the data. We use some data to calibrate the model, and then use some data to really validate our model. So what you are seeing here is the validation result. And again, you will see that the scatter plot really indicates that the observation, which is the x-axis, the simulation is y-axis. These observation and the simulation are really matched well with each other. And then these are from the validation, basically the auto sample prediction. And then so again, this achieves a high confidence that our model works in terms of simulating the traditional corn soybean rotation system of our landscape in the Midwest. And then so that study has other details, but I think I will skip that. But with that study that really demonstrating, we have a lot of confidence that we believe the model can simulate corn soybean rotation system. Now, the next question that we have is, can we also simulate cover crop? Because in the third study, we haven't really demonstrating cover crop implications. So then we go to the fourth one, which is the cover crop. So for the cover crop one, what we did is we look at the 7 sites that Professor Marion Villamil collected over the whole, across the state using the NREC previous funding. So what she did is she conducted the trials of cover crop trials. These are the different sites, locations, latitude, rainfall, these are conditions, including some of the soil organic carbon storage. So these are the ground truth data that, Professor Villamil collected over the past few years. And then she has published some papers, but these data sets very valuable. They have never been really used for the modeling approach. So we take these data, collaborating with Maria, and take these data, really try to understand, can we really simulate the biomass of the cover crop, and can we really make sense out of that? And so what we did again is we look at our, in these systems, it's the real production system that you grow cover crop, but you also grow cash crop in the summer. And then so the trial experiments take all the biomass of the cover crop as well as the cash crop biomass and the yield. And so we are able to validate both cover crop as well as the cash crop results. And in this particular result, what we're showing is the results that we do have in the simulation can largely will match the cash crop prediction, cash crop production, in terms of the corn and the soybean. And these are the scatter plots. We basically compare our field level measured yield versus our simulated corn yield. And then, this is a typical, cover crop sites that from Marian's previous trials. And then one major implication from the, after you validate the result, we have more details of the validation of the result, but it depends on your need. We can go into there. We also validated the biomass information of the cover crop. I don't have a slide here, but we have very rigorously validate the biomass of the cover crop too, because Marian measured this. And then so they also achieve a very high confidence. Let me see whether I can quickly pull out a result here. Give me a second. Let me see, let me stop sharing a little, pull out the right figure here. So we also had a very, very detailed validation of the biomass information of the cover crop. Give me a second. And so this result provide, these are the different, okay, I come back to the presentation. So this is the cash crop validation at the size scale. And then we move to, this is the biomass for the cover crop. And what you are seeing here is the x-axis, the measurement, the y-axis is the cover crop biomass. And then you will see that we actually look at the different cover crops. We look at ryegrass, we look at cereal rye. Ryegrass is in blue, cereal rye is in green, and then hairy veg. And then so you will see that all these are different cover crops that collected over the years by Professor Villamil sites. And then these red curve means one-to-one line. So you will see that again, our simulated result, which is in the y-axis, the observation from the trials is in the x-axis. And again, they are very much corresponding to each other with a very high R-square. And so these results provide a sufficient confidence that we not only can capture the biomass of the cash crop, but also the biomass of the cover crop. And then so with these understandings, we start to see that, hey, what if we start to grow cover crop in the coming few years? What will be the implication for soil organic carbon? And in this case, we are looking at soil organic carbon implication. So the x-axis is the different yields. So from 2013 to 2020, we're simulating the path of the dynamics of the soil organic carbon at the whole zoo zone from up to 1 meter. And then what we are looking at is each curve refers to basically the time series that you simulated in the model with cover crop versus without cover crop. So the blue refers to the curve that you don't plant any cover crop. And then the yellow refers to you grow cover crop, but that's a typical rotation of corn, followed by cereal rye, and then soybean, followed by hairy veg. And then the red refers to more tradition, another cover crop kind of rotation. We grow corn, soybean rotation, but in the middle we grow ryegrass. And so if you look at these three scenarios, what you are seeing is the blue refers to without growing any cover crop. You actually see that they actually reduce the soil organic carbon in a more dramatic way. But then if you start to grow cover crop, you still slightly reduce your soil organic carbon, but then the benefit is very clear because you can see the difference between these two curves, red or yellow versus the blue. So you will see that the difference between these two basically give you the ideas of what's the benefit of growing cover crop in terms of maintaining the soil organic carbon in the soil. And then, I don't think we need to really explain the benefit of SOC, soil organic carbon. We hope that our soil can be rich and then maintaining the soil organic carbon. And then we demonstrate that cover crop really help you to do that. And the benefit of doing, using models is, in reality, if you grow cover crop, usually you grow 3 years of trial. But if you only look at 3 years, the difference is very small. But if you calibrate a model, you make sure your model works, then in the modeling world, you can start to really simulating longer time, like 10 years or even 20 years. And then by doing this, once the model become convincing, has been fully validated, you have confidence that what you are seeing in the modeling world actually makes sense. And then, so this is one of the take-home message that we have. But at the same time, in the model, we also do something extra, also very interesting. So for example, in this particular case here, what we are saying is, let's take a look at the ryegrass biomass. And then if we plant the ryegrass biomass at different date, so these are like different 4 starting dates that you put the biomass in for ryegrass, what the biomass you can gain. And what you actually see is that if you plant biomass, ryegrass, earlier, you can gain higher biomass compared with, later planting of cover crop, you get lower biomass. And another very interesting implication that people will care about is when you terminate cover crop. If you terminate cover crop too late, you will actually, so these two are talking about the termination of cover crop, and above two talking about the starting, when you plant the cover crop. So when you plant the cover crop, if you plant the cover crop earlier, you actually gain higher biomass. But also there's a consequence that if you planted it relatively too early, you actually may get a little bit yield penalties. And these are the hypothetical numbers. Just don't really think this. This is more like we are trying to show that in the very ideal world, if you plant cover crop too early, then you will have some yield penalties. But then on the bottom two, that shows the cover crop termination time. If you terminate cover crop earlier on, in April versus April 30, your cover crop biomass will be reduced. But also that reduction probably can benefit your cash crop yield. So this is again, is the cash crop yield. If you terminate the cover crop early on, you have less impact on the cash crop. And the reason is mostly because if you let the cover crop grow too long into this, terminate too late, then cover crop will become a competitor of water resources as well, nitrogen in the soil with the cash crop. And then so some of our modeling studies start to really help you infer the results of how you really manage cover crop in terms of when you terminate and when you harvest, when you plant and when you terminate, sorry. And then, so with all these in mind, I think that gives pretty comprehensive ideas of what we have done, these 4 studies. And definitely there's an aspect that we need to continue to improve, but that should give you a brief idea of what we have been working on. And then, so I think I will, and then the next 2 things we really hope to ask our committee's guidance is, first, you please raise questions about any studies that I presented, but also what I really hope to learn is, I'm a modeler, so I'm a modeler, I'm using satellite, I'm not doing trials at this moment. So I'm, relatively not a typical way of doing agricultural study, trial study. But I also have a very deep, I think I have a very deep belief that when we use models and properly use model and satellite, especially when the model can be validated with the trial data, the model can really be helpful too. So I have that belief, but I think how to best use this information and what extra things that our stakeholders like to see. I really want to open that up and solicit suggestions and feedback. And this is one aspect. And the second aspect is, let's say we have these papers finished and published, how we really communicate effectively with our stakeholder, whether it's through a website or through a presentation, or through a field day demonstration or some other aspects. I really like to have this discussion. And I think, so the next 20 minutes, I really want to devote to that. But before we start that, I also want to say, use 2 minutes to talk about, this project is what I submitted as potentially, I submitted this year. It's not get funded, but I think it's a natural kind of a continuation of what I hope to do for under the NREC project. So I will continue submitting this proposal next year. But what I really hope to do is, we know that the MRTN tool for nitrogen advisor, and then these MRTN tool previously developed by Professor Emerson Knifesinger, actually continuously we collect more data over the years by Dan Schiffer and Professor Emerson Knifesinger. And then these on the right side, it shows you all the data sets that we have collected by the NREC effort. It's a rich data set and a lot of data. And then what Professor Emerson Knifesinger really hope to continue is how we can take these newly generated rich trial information with the MRTN tool, because the current MRTN tool is really based on the outdated results. And then so can we build a specific MRTN tool for our Illinois farmers? And then so this is the motivation that we try to see, can we leverage the existing invested results? NREC already invested a huge amount of money to conduct these trials. Can we leverage these results and build into the MRTN that we can really build a specific tool for Illinois farmers for the MRTN? And this version can have the classic version that Professor Emerson has been done. We're gonna keep that version, but also there's a possibility to bring some of our new modeling capability to have some more intelligent version. And so we believe that providing both information openly, freely to farmers would really benefit Illinois farmers. And this is a project that we are really motivated of doing. Hopefully we can get support next year, but even without support this year, we already start to build some efforts. And I want to keep the NREC committee informed about what we have done without any support from NREC for this specific part, because that's where my passion lies. And then so basically to summarize, I think we want to clearly wrap up the current cover crop project. There's still space for us to add more analysis and modeling. We want to collect feedback. We want to really collect the feedback from our committee stakeholders about how to best communicate our result with our stakeholders. And I think, I have said enough. So I really want to use the rest of the time to collect the feedback from our committee members. Thank you.
Thanks. Hey, Kaiyu, do you want to introduce your team member there too?
Oh yeah, we only have, I think we only have, let's see, how many team members we have. Oh, I only have a student, Yi, so yeah, he's one of the graduate students that are working on the project, so no worry, yeah.
Okay, just wanted to make sure we didn't leave them out.
yeah, I really appreciate that. So yeah, let me get back to my presentation. Maybe somebody can get started, and I really want to, Yi and I gonna take notes, and we really like to hear feedbacks, including the current research. Do you think the current research makes sense, or you think there's a lot of ways to improve that? And all these questions are really worth, really gonna be very meaningful and helpful for me.
So Kaiyu, this is Jeff. I'll go ahead and start with a few questions that came to mind from a farmer
Sure.
perspective.
Sure.
So it's really interesting and exciting as I watched you go through this, because I think these are uncovering things that farmers always have questions about. Some of the work that you did, the nitrogen, NDVI or however your chlorophyll measuring, does that have any integration of the cover crop with it, or is that just purely nitrogen on corn without the cover crop involved in that? And then if it's not, do you have the envision of including cover crops into that, and how that changes our nitrogen models, how we address as farmers the carbon penalty, if you will, or whatever in that nitrogen scenario for, I mean, that's what I think I'm looking for, a solution in nitrogen management in the presence of a cover crop that we've terminated, and kind of guessing how to manage that.
Yeah, Jeff, really appreciate that question. To a certain extent, because I mentioned I'm not doing my own trial, so to a great extent, we a little bit opportunistic in terms of what trials we can really monitor. And then so in this case, this is a long-going trial by Emerson in the South Farm, and he's only currently doing traditional nitrogen trial. So in this study, we only did the nitrogen studies without considering cover crop. Now, if there is an opportunity that somebody in NREC or other studies in the campus nearby that doing a combination of both nitrogen and cover crop trial, we really like to know, and we want to team up with the person to bring in the component of the monitoring sensing component, because for us, bring that component is not difficult at all. We just need to know where and when you are doing what. And then
sure.
Pretty much, if we want to fly drone, we can design to fly drone. If we want to fly, make sure we get the satellite data. In reality, we don't need to worry about satellite data, because satellite data is continuously capturing all the time. So we just need to know, where the location. We just go to the data archive to grab the relevant data, and then so depends on what kind of data we want. All the NASA satellite data are free. So they free to, all the citizen, actually NASA is open to the world. So it's all free to everybody. And then if we want to use some of the commercial satellite data, like, Planning Lab, our university already have a pretty good license. So I don't need to pay, NREC don't need to pay. And then we just grab the data and do the study. So I would say that it would be very nice to develop some project that has both components of cover crop and nitrogen trials. And I see that if that's a need, I really want to, basically see whether there's an opportunity that I can talk to Emerson, I can talk to Marin to see whether they have interest in developing a specific targeted trial study. And then I bring that component in. I would say that these are very important, if especially farmers, especially our producers, stakeholders think that's an important question. I believe we should open up to the research community and see, this is important and we should do it. And I'm gonna sign up for that to bring my sensing component in.
So Jeff, really quickly, I want to address your question because it's going to lead to my question.
Okay.
So the whole concept of remote sensing platforms for guiding or directing nitrogen inputs into a corn production system is we're allowing the plant to integrate all of the information and the environment to which it's exposed. So presence or absence of a cover crop really shouldn't necessarily matter. While saying that, there might be, if you have enough observations, you can actually come up with, there's a lot of different ways, a fudge factor, a back-off coefficient,
right.
some sort of a coefficient that if we know that we're traditionally biased by the absence or presence of a cover crop, maybe we adjust the model. But the whole concept of remote sensing platforms is you're trying to let the plant integrate all the information, and you're just simply making observations and responding based upon some model that you develop to generate a recommendation.
That's
so
right.
That leads me to my next question, because one of the bigger challenges associated with use of remote sensing platforms is the traditional NDVI approaches that we use have limitations. There's sort of a window of opportunity where they work really, really well. And outside of those windows, you have some challenges. And one of the big ones is how early can you detect differences not only from some of that's driven by the physics of how the sensors function, and it's also driven by how much information have you ultimately collected about that crop when it's only 35 days old? What is ultimately going on in that soil, and then how well can you predict what the environmental conditions are going to be like after the time of the sensing? So I mean, it's the way we use it is sort of simplistic, but there's a lot of complexity to actually using those sensors to make decisions. So my question was, Kaiyu, specifically for you,
Sure.
are you guys evaluating, or whoever you're cooperating with, are they evaluating different? So we're traditional. I'm an NDVI. I grew up with NDVI. I grew up with simple ratio. I grew up with inverted simple ratio. And you can use, NDVI doesn't necessarily just mean one thing. NDVI can be a green NDVI. It can be a red NDVI. You just have to pick different wavelengths. Have they identified, and I haven't kept up with this, to be honest with you, have they identified specific wavelengths that may work a little bit better earlier than the traditional red NDVI where we're using the ratio of red plus near infrared divided, or actually red minus near infrared over red plus near infrared? Are there any changes in that? I haven't kept up. I apologize.
Robert. So what I'm going to show you is, and again, the research that conducting at my lab, we are doing more sophisticated hyperspectral stuff and different remote sensing stuff. And so your question is exactly, here. So we try to interpret, if you use hyperspectral, so instead of only NDVI is in red band and the near infrared band, and you put them as an index. But if you start to look at the whole wavelengths from 300 nanometer to 2500 nanometer, and then if you slicing
it, sure.
And then so you actually can reveal way more information and then capture way better kind of nitrogen content, chlorophyll, as well as photosynthetic capacity. And our lab is actively doing that. And then to a certain extent, we haven't really talked this much in the NREC community, mostly because I feel that it's a challenge for me to really bring very, very advanced study to the committee, because I'm worried that it's not easy to communicate, and that's my challenge. Modeling itself is very complicated already, and then if I telling that there's a cool sensing technology, but then if there's a I do have a challenge here that to communicate. So for example, we actually done a lot of work here, like looking at nitrogen trials, and these are all the trials in Champaign. All the nitrogen trial, we use hyperspectral. We actually fly airplane of the hyperspectrum. We get a lot of exciting stuff, like understand the nitrogen and then bring understanding about can we really map out nitrogen. So let's see. So this is a graph that if we fly over hyperspectral, we get rich information, not only as broadband NDVI, but 300 bands in the whole wavelengths.
Right.
And then by doing so, estimating nitrogen we start to become really, really confident. And then this type of the technology potentially can be benefited by farmers, but also because this technology is still in the very early exploratory stage. We feel that when we feel the technology become more mature and then ready to be disseminated, then we see maybe that's a better place for NREC. We do have that, thinking going on. And then so, so this is also why when we bring in projects, we really want to bring in projects that we believe there's information that we generated that can be disseminated. And then this information we couldn't disseminate because we only have limited funding to fly over some trials, and that's it. And then so we can publish papers, but my understanding is that I think NREC will be better. NREC's investment will be better paid off if we have something that farmers can directly tap into. And then so that's kind of coming to my question that like, what's the best way to communicate these results? And then Julie and Jeff, Robert and Jeff, I may have an excuse because I have a sharp stop at 11 a.m. and because I have another teaching commitment, kind of a guest lecture thing, just one time. But I want to see that I would like to ask Julie whether I can have another opportunity to continue this discussion, because I feel that I haven't gotten enough suggestion yet. I want I want to Robert, Jeff, and Austin to tell me more instead of I'm talking. I want to set this up, and then so before it's kind of getting 11 a.m. too late to see this.
Yeah, we can definitely. I mean, from Jeff's perspective, where we probably have a fairly short window before he starts harvest.
Yeah.
But I can send something out, and we may even be able to rope a couple other folks in if they have an opportunity to watch this part, and then we could have a follow-up. But I would also say, Robert, Jeff, Austin, if you guys have thoughts, go ahead and shoot an email. I'll make sure you guys have Kaiyu's email. And I think I have Zia. I think I have your email as well, so we can copy you on that. And then, so we can continue the discussion for sure.
Yeah, I'm proposing that we have another meeting, in the coming few weeks before, especially before Jeff starts to be extremely busy in harvesting.
Too late. Jeff's already busy.
Yeah.
I know you're always busy.
Just level of degrees of busyness.
So I think my window is pretty open. I don't harvest, so I don't harvest, and then so we actually happy that field work reach to the end because we only capture growing season. And then so Julie, I think it will be really nice to set up another meeting next week, or whenever you feel most appropriate for our stakeholders and committee members. I really want to have that communication to collect wisdom and guidance about how I can best communicate and what kind of research really suited for NREC, because that's important to me. I think
Yeah, we can do that. I can send something out and see what folks' availability is next week.
Yeah.
See if we can get it set up.
Yeah, and I will just I want to follow up with this. Make sure Jeff and Austin and Robert, if you can think about more questions, especially questions that can help me. What are some burning questions that, for example, Jeff, you as a producer, you care a lot, but you don't find the answer, in particular regarding cover crop. And then likewise, Robert, you being in the industry, you are the expert in the industry. If there's anything that we can potentially help a little on that side, and I think the broader audience for the NREC program includes both farmers as well as industry. So I wanna, and I think the goal for me for the program is I want the study that funded by NREC at my lab to be really have a public goods component. So we release all the insights. If we generate useful tools, we want to open source that and let everybody use it. And then so I think that's also we did not have time to cover that today, but I want to have that in our next meeting for detailed discussion, like what kind of way to best communicate and disseminate.
Perfect.
Okay.
Yep, you better get to your call. We don't want to make you late.
I appreciate that. Thanks,
All right.
Jeff,
Thank you very
Robert,
much. It's really informative.
Really
Thanks, everybody. I'll
Thank
send out a note to find a date, guys.
appreciate that.
Thanks, Julie.
All right. Thank
All right,
you.
thank you.
Thank you.
Bye.